Hey everybody, welcome to Barton's Biology. Today, DNA replication. Last time was DNA structure, a little bit on how DNA is put together. And today, kind of how it's pulled apart. More on that soon. First, what is DNA? Remember there are 4 nitrogenous bases, and what are those 4? And what does DNA stand for? Give yourself some time, pause here if you need to. Remember, this is a video, so you can pause, rewind, fast forward, whatever you got to do. First, what are the 4 bases? Those 4 bases you see here in this digital model, they are adenine, thymine, cytosine, and guanine. Remember that adenine only pairs with thymine, thymine only pairs with adenine, and cytosine and guanine always pair together. DNA stands for deoxyribonucleic acid. Deoxyribo is all about the deoxyribose, the sugar that's in the nucleic acid. Okay, so sometimes because your cells need to reproduce, or a bacteria needs to reproduce. These are some of the fundamental characteristics of living things. These things need to reproduce, and for that to happen, the DNA needs to be copied because every cell needs to have its own copy of DNA. Those are the instructions that tell that cell what to do. So that replication happens in a really cool and efficient sort of way. First, step 1, a helicase enzyme. This is just a protein that comes in and pulls apart the 2 strands of DNA, pulls apart those 2 halves of the ladder. It breaks the hydrogen bonds in between, and each strand, each half, is able to separate. Okay, just like here in the picture. Then, helicase, after helicase comes in, unwinds the DNA before replication, we get a second enzyme. And that second enzyme, in this case, is called DNA polymerase. DNA polymer ACE, right? Polymer, because DNA is a polymer, it's a macromolecule, and those are all macromolecules. It's made up of small parts called nucleotides. If you don't know what those are, go back to that DNA structure video. So here, DNA polymerase reads the old DNA strand, and that strand serves as a template to create new bases, to create a new strand on top of. Okay, what I mean is if we have, for example, right here, an adenine, that means the DNA polymerase, the green molecule here, will put a thymine right next to it, or vice versa, right? So if it's a thymine, then it'll put a adenine. If it's a cytosine, it'll put a guanine, okay? It reads the old strand like a template and builds the new bases in complement to the first strand. Okay, polymerase is just an enzyme that builds the new strand that is complementary to the DNA strand, and the hope is that we don't get any errors. But sometimes you can see, just like in this image here, sometimes errors do exist. So what's the result of all of this? Well, hopefully, at the end, there are 2 completely identical molecules of DNA after this process is done. Okay, each of those strands of DNA contains 1 old and 1 new strand of DNA. Each one of those DNA molecules contains 1 old and 1 new strand, and we call this process semi-conservative. You can see that there. Here's another image, just to kind of help you out, maybe kind of visualize it a little bit more. You can see the helicase splitting the DNA apart, the polymerase building the new strands, putting new bases on. And that, in a nutshell, is DNA replication. Anyway, like I said, if you don't know those parts and bits and pieces of DNA, go back and review the DNA structure video. If you do, rock on, you're going to do just fine. Have a great day. I'll see you later.